questions. Thanks all again for coming out on such a splendid night. Professor Yin, we ask a few questions like, where were you born?
I was born in Kunming, China.
Kunming, China. And where'd you go to high school?
Colorado.
Where in Colorado?
Aurora, Colorado.
Aurora? Oh, my aunt lived in Englewood.
Wow.
7120 South Uinta. And where'd you go to undergrad?
Princeton.
And graduate schools?
Michigan.
My condolences. Sorry, I can't help it. And how long have you been here?
39 years.
Wow. Let me do the math. How long ago was that? What year would that be then?
1977.
Wow. Okay. And you just retired?
I'm retiring at the end of August, yeah. So soon.
When is the party? Okay.
I don't know.
I just want to point out, this is one of the great things about this. When I got the email that Maya couldn't do this, I sent out some emails, and it's pretty extraordinary to be able to have someone like Professor Yin as the secondary speaker for today. So would you please join me in welcoming Professor Yin?
Thank you, Tom, and thank you all for showing up on a pleasant summer evening when I'm sure you probably had some better things to do at home, but it's nice to see interest in science, and it's I'm struck by the incredible diversity of animals that are being studied here, right? So I'm going to talk to you about the general problem that I studied for these 39 years, although actually, you know, I only really touch a little bit on the experiments that we do. But the general question that we're interested in is how does the brain put together information from our two ears? Right? And this is analogous to, I think, the probably better known question is, why do we have two eyes? Right? So most people, when asked why we have two eyes, would say, well, it gives us vision and depth, right? Gives us stereoscopic vision, which we can experience when there are 3D movies showing. And you can demonstrate that, you know, relatively easily. In hearing, I think there's a very analogous reasoned, you know, answer to that question. Just as having two eyes gives us another dimension, hearing with two ears gives us dimension of spatial hearing. And we'll all argue about how the visual and the auditory systems are different, and therefore, in some ways, this question is more, at least, is of interest because the brain is making these computations. So the you know, answer to the question usually is given that hearing with two ears allows us to localize sounds in space, which is true, and that's basically what I've studied for many years. But as we'll see, there are some subtleties to that, which are, I would say, probably counterintuitive and probably not widely known, which I hope you'll find interesting. So if you think about sound localization and its importance, you know, for humans, we've probably kind of evolved past the point where being able to localize sounds is crucial to our survival. Unless we happen to be in London and we step off the corner, you know, the curb without paying attention and forget that the traffic's coming from a different direction. But if you think about just about any prey or predator animal, you know, if they hear a snapping twig on a dark night in the woods, their survival may depend upon being able to localize whether that's a predator that they have to be wary of, or whether that's a prey, and if they're hungry and want dinner, they have to be able to localize it. Right? So as a result, there's a considerable part of our brains that are devoted to this task of integrating the information from the two ears. So I want to start with a video which illustrates nicely, I think, how well animals can do this job. Right? Let's see.
It listens for the tiny sounds of its prey moving about below, but must take great care not to scare them away.
So how do we do that? How does the fox do that, right? Remarkable. Because probably if he's a couple inches off the bowl or whatever it is he's hunting, gets away, right? I wish I could tell you that this is the behavior we study, but even though we have lots of foxes and plentiful snow usually here in Wisconsin, I'm going to talk about my lab has also done some behavioral experiments. I'll show you briefly, but it's much less exciting than that one. Okay, so let's just briefly talk about differences in localization in the two major senses, vision and hearing. So in vision, and just like this, similarly in the other, I would say the third major sense, which is touch, right, somatosensation. In both vision and in touch, the external world is mapped onto the sensory epithelium, right? In the case of touch, onto our skin. We know we're touched here and not here because the receptors are activated when we touch that point. Similarly, in the visual system, there's a map of the external world mapped onto the retina. Let's see, I think I have it. Well, do we have another pointer?
Pointer.
Maybe this is just, yeah.
Use the cursor on the
Oh,
on your computer.
yeah, okay. Anyway, so as sort of diagram here, find the pointer. The retina has a map of visual space, right? So the bird that's in the visual scene there projects onto a spot in the retina. So in the brain, the problem of telling where that bird is in space is a matter of keeping track of that part of the retina as compared to the rest of the retina. So we have a retinotopic representation of the world on the retina, and then in every one of the relay stations on the way up to the visual cortex and beyond. Right? Now that map can be distorted, so central vision has a much larger representation than the peripheral part of the world. But nonetheless, that problem of visual localization is a sort of an anatomical problem. Here's I don't know if it's the switch or the battery. Whereas in the auditory system, on the sensory epithelium, there's not a map of space, but rather a frequency. So what the cochlea does, and I'll show you a brief video to try to give you a sense of that. What the cochlea does is to break down the sounds that we hear into its frequency components. So those of you who might be engineers or physicists, this is very much akin to Fourier analysis. It does a Fourier transform of the sounds and represents that along the length of the cochlea. So auditory space is not represented in the ear at all, right? So therefore, in order to localize sounds, we have to depend at least partly on the very small differences of the inputs to the two ears, right? And then the brain has to put that together, right? And so that's the question that my lab has been working on. How does the brain do this? Well, the two major cues that the brain uses are interaural time differences, shown on the left, and interaural level differences, shown on the right. So, for example, a sound source off to my right will reach my right ear slightly before it reaches the left ear, right? But of course, sound travels pretty fast, and our head is relatively small, so those time differences are very small, right? The maximum time difference in a human is less than a millisecond, right? And if I were to put headphones on you and play identical sounds with just some small difference in the timing of the sounds to two ears, the just noticeable difference is actually a remarkable less than 10 microseconds. Right? And I'll give you an example of how remarkable that is. The other major cue is the level difference. So a sound off to the right will also be louder to my right ear than it is to my left ear. That's kind of what's schematized here. So therefore, we can use those differences in the loudness to the two ears as a cue. Right? Now those two cues actually are used in different parts of the frequency spectrum. So obviously, if we're going to detect timing differences, then the ear, the cochlea, has to encode that timing information, right? We're going to compute the difference between the right ear and the left ear onset of the sound. The cochlea has to somehow be able to code that timing information. And that timing information is coded by so-called phase locking of the auditory nerve fiber, well, the hair cells and then the auditory nerve fibers. And I'll show you a video in a moment to give you some sense of that phase locking. And we know that from experiments in many animals that phase locking only is present for relatively low frequency sounds. And here, when I say low frequency, I mean frequencies below about 3 or 4 kilohertz or so. So therefore, as expected, ITD sensitivity is really only best for those low frequencies. And I would argue that this is kind of the premier example of the ability of the brain to detect minute timing differences, right? Remember, the J and D is around 10 microseconds. So here's a video of recordings from the auditory nerve of responses to a 400 hertz tone. This is just mainly to give you a sense of the way the nervous system communicates with action potential. So what you'll see is a oscilloscope trace with recordings from an auditory nerve fiber, and it also illustrates the phase locking. So here are the action potentials, or spikes called. This is the way in which neurons, all the neurons in the brain pretty much, communicate with other neurons, right? So they use these all or none electrical pulses. And I hope you can hear when the sound is delivered, right? Well, on. And this shows you that when you trigger the trace at 0 time here, the next spike tends to occur right around here, which happens to be 1 over the reciprocal of the frequency of the tone, which was, in this case, 400 hertz, right? So that's what we mean by phase locking, is that the peripheral, the auditory nerve, tends to respond at a particular phase angle of, in this case, the input sine wave. Okay? And this is just an illustration to sort of hammer home the remarkable timing sensitivity of the auditory system. So we all have in our mind approximately how long 1 second is, right? So divide that 1 second interval into 1,000 parts, right? That's a millisecond. So this is a millisecond, right? On this illustration, this is kind of an action potential like we just saw in that movie, right? Now divide that 1 millisecond into 100 parts, and that gives you approximately, you know, I've tried to illustrate it here, that's approximately 10 microseconds, right? So our auditory system, our brain is able to detect a 10 microsecond difference. That would be a sound that's just off the midline. Right? And psychophysical experiments in humans show us that human subjects can detect a sound that's about a degree or 2 off the midline. Right? Remarkable sensitivity. And it turns out if you look at different animals, so in general, predators have much higher resolution, be able to resolve these very small timing differences, probably because in order to get that lunch, you know, they have to be able to localize exactly where that prey is. If they're off by 10 degrees, they're probably going to miss, like the fox, right? Fox is off by a couple inches, no meal. The vole or the rat, on the other hand, right, just has to know, well, did I hear that on my right or to my left, and then scamper the other way. In any case, this is you know, kind of what got our lab started, in the interest in being able to encode those time differences. And I'll show you some examples of cases in the nervous system where it looks like the brain has become very specialized exactly to deal with this problem, to be able to resolve very high timing differences. The other cue, internal level differences, depends upon another way to think about that difference between 2 ears in level, is that the head has to act as a shadow, right? It has to shadow, it has to attenuate the sound to the far ear with respect to the near ear. And it turns out, just for physical considerations, this can only happen when the wavelength of sound, of that particular sound, is shorter than or about the dimension of the head. Right? So longer wavelengths, the head doesn't act as an effective shadow. I'll show you an example of that in just a moment. And again, the difference between low and high frequencies is around 3 or 4 kilohertz. Right? So above 3 or 4 kilohertz, ILDs are an effective cue. Below it, they aren't. And so therefore, we expect ILD sensitivity to be primarily a high frequency phenomenon. And here's a sort of intuitive drawing of why the dimensions of the head play a role here. The dimensions of the head relative to the wavelength. So if you consider waves in water, which are really like the waves in air that we hear sound, you know, if we have waves in a lake like this, and let's say we had a battleship sitting out there, right? Then obviously those waves are going to be attenuated on the other side of the battleship, right? Whereas if we just have some reeds in the water, though there will be virtually no attenuation because the size of the reeds are much smaller than the wavelength of sound. Right? Okay, so now I'm going to cheat a little. So I teach this material, and I found this really nice video which summarizes what takes me about 30 minutes in lecture into about 2 or 3 minutes. And this is actually done by a fellow who was an auditory physiologist who became
Audiences developed a voice processor chip that provides advanced
technology
noise reduction for
specialist,
cell phones
and
based
he
on the
used what he
operation
knew
of the
about
human hearing
auditory
system.
system
By
develop
adding
something
audience's
that's
voice
probably in all of
processor
your
and a
cell
second
phones.
microphone, we give your cell phone the ability to hear with 2 ears, thus making intelligent decisions about preserving your voice and suppressing background noise. Let's start by taking a look at how the human hearing system works. The first part of the human hearing system is called the auditory periphery, which consists of the outer ear, middle ear, and inner ear. Sound waves in the air around us are captured by the outer ear, funnel down the auditory canal to the eardrum, which causes the 3 bones of the middle ear to push in and out on the cochlea. If we could unroll the cochlea, it would look like a long tube filled with water, with the basilar membrane running down the middle. The pushing by the last bone of the middle ear on the cochlea causes waves to travel down the length of the cochlea. These traveling waves in the cochlea are sensed by 3,000 inner hair cells arranged in a line down the basilar membrane, with hair cells at the front of the cochlea picking up high frequencies and hair cells at the end of the cochlea picking up low frequencies.
So this is the frequency
Each hair
map
cell has about
across the
10
cochlea.
auditory nerve fibers, so the 3,000 hair cells convert their signals to spikes on the 30,000 fibers of the auditory nerve, creating a rich spectrotemporal representation of sound. Here are some examples of interesting sounds. Tones. Transients.
So this is kind of
Pitch.
a Fourier transform of sounds
Formants.
by the cochlea.
Speech. Audience voice processor. Music. Now that we're familiar with the computations done in the cochlea and the signal carried on the auditory nerve, let's take a look at the rest of the hearing pathway in the brain. The auditory nerve projects into the cochlear nucleus in the brainstem, which is the oldest part of the brain at the top of the spinal cord. Here there are 5 major cell types that compute derived representations of the sound, each specializing on different aspects. For example, the octopus cells respond only to broadband transients like this. These signals from the cochlear nucleus project to higher centers within the brainstem. The superior olivary complex is the first auditory brain region that receives signals from both ears. The medial superior olive computes time of arrival differences, and the lateral superior olive computes loudness differences between the 2 ears. These two...
Okay, I'm going to stop it there because that's essentially what I'm going to talk about in just a moment. So just to give you a better sense, here's a scanning EM of the cochlea. You can see the hair cells. There are 2 kinds, inner hair cells and outer hair cells. There's a single row of inner hair cells and 3 rows of outer hair cells, and they're spiraling around. In the human cochlea, we have about 3.5 turns, and there's this map of frequency along these 3,000 hair cells. And these hair cells are the device that transduces the mechanical energy that is delivered by the sound waves into electrical signals that the nervous system can understand. And it's obvious that these hair cells have been designed for high sensitivity, right? So in other words, the system has been designed to be able to detect very faint sounds, right? So at the threshold of human hearing, the movements that would be measured in the hair cells and in the basilar membrane, the membrane that these hair cells sit on, those movements are on atomic dimensions, right? So it's estimated that they wave at threshold. They move at about the width of a hydrogen ion, right? So they're about as sensitive as you could possibly make them. The flip side of that is if you're an engineer, you design for one thing, you sacrifice something else. So we've designed this cochlea for sensitivity. What we've sacrificed is it's vulnerable to damage by loud sounds, right? So if you turn up your iPod up to maximum volume and walk around all day with that, these hair cells will degenerate. And once they degenerate, they're gone for good. They don't regenerate.
Right.
So most hearing loss results from damage to the hair cells. And in our modern society, a lot of that is self-induced, so you know, at least man-made sounds. If you think about it, you know, in nature, there are not very many loud sounds, right? Or if there are loud sounds, they're very transient, you know, clap of thunder, you know, the roaring of a saber-toothed tiger at your cave can be loud, but then you've got other problems than hearing. But in modern society, what are our loud sounds? Well, factories, guns, iPods, you know. So they're all, so this is just a cautionary note to turn those volumes down. So here, again, these hair cells are wonderful cells. So essentially what happens is when the mechanical movement of the tympanic membrane sets in motion the fluid, these are all setting in fluid, and that fluid causes the hairs, they're called hair cells because they have these tiny hairs, these hairs to move back and forth. And when they move back and forth, here's a blow up of one row, these are called stereocilia, one row of stereocilia, and you can see they're arranged from low to high, and they have this little tip link, and it's now thought that that tip link is really the transducing element. So the general model looks like this. If you push the hair bundle in the here towards the right, then it stretches this tip link and actually mechanically opens an ion channel just like a trap door would open. You push it back the other way and it closes that channel. Right? And this mechanical gating of the channel makes the hair cells very fast. They don't depend on some chemical second messenger system like we have in the retina, for example. Right? So that allows the auditory system to respond very quickly. Okay, so as that kind of as an introduction, let's talk about, you know, binaural hearing, hearing with two ears. So these are some examples of devices that have been built to hear localized airplanes, and here another device. Note that this one has 4 ears, 2 on the sort of horizontal dimension and 2 on the vertical dimension. And we'll see that that's an important consideration. Localizing horizontally and vertically are different. Right? And if you really want to eavesdrop on the conversations in your next cocktail party, you know, this is a good way to do that. And here's just, you know, if you really want to experience this, you can actually go to Germany and sit on the throne, hearing throne. Okay, so let's talk about a little bit about the physiology and anatomy that accomplishes this task. So the auditory nerve fibers come into the brain and they synapse in the structure known as the cochlear nucleus. And we have one on each side, right? They're right inside as the auditory nerve comes in the brain. And immediately, every auditory nerve fiber bifurcates, with one branch going to the so-called ventral cochlear nucleus and another branch going up to so-called dorsal cochlear nucleus. And on the way, you can see they encounter a bunch of different cell types that was alluded to in that video. Different cell types, and they have funny names because anatomists like to, you know, describe them. So they're so-called bushy cells and octopus cells because they look a little bit like an octopus, and fusiform cells. And here's a sort of schematic of that. And kind of the remarkable thing is, you know, the information coming in here in the auditory nerve is the same to each of these cell types. But it turns out if you record from these cell types, you actually see that they're quite different. So here are just a schematic diagram of 5 of these different cell types. Here's sort of the response you can imagine that you heard on the video of the space locking, right? So if you deliver 100 of these and sum them based on the onset of the stimulus, you get something like this, right? The tone is on here, and it responds during that tone. If you do the same thing as you're recording from these other cells, you can see that some of them are very much like the auditory nerve fiber. And in fact, these responses are termed primary-like because they're like the primary afferents. But others depart. In fact, the extreme one is a cell that only responds at the onset of the tone. Remember, those are the octopus cells that were alluded to in that video. Right? And this has been described by many investigators. Joe Adams was probably one of the first, and shows you the variety of cell types in the cochlear nucleus. Now, the ones that are of interest to us are the bushy cells. And it turns out that the nuclei that do these computations of ITDs and ILDs, both of those rely on bushy cell inputs, right? So just as I've sort of set up this sort of straw man that, you know, we're sensitive to ITDs at low frequencies and ILDs at high frequencies, it's not surprising to find that there are actually two circuits, one that computes ITDs and one that computes ILDs. So the ITD circuit involves cells in the medial superior olive that get input from the bushy cells on each side, and the cells that are sensitive to ILDs are in the lateral superior olive here that get excitatory input from the bushy cells on one side and inhibitory input from the relayed through the bushy cells on the other side. So these cells in the LSO are sensitive to the relative level of sounds because they get excitation from one side and inhibition from the other. Right? Now, this you know, absolutely simple looking circuit is the one that we think is computing the ITDs. And the remarkable thing about the cells is that these cells in MSO respond as if they're coincidence detectors. That is to say, they respond maximally when the inputs on the two sides are exactly coincident, right? And if you time shift them slightly, they stop responding. This is just an example of one of the specializations that I talked about. So here in that circuit that's computing ILDs, we have a synapse from the bushy cells on one side to this nucleus called the medial nucleus of the trapezoid body. And those synapses are shown here, examples of different synapses. These are known as the calyx of Held. Held was a German anatomist who first described them. And these are widely called the largest synapse in your body. Right? So here in this circuit, the nervous system has developed this very, very specialized synapse. There's virtually no other synapses in your brain that look like this, certainly in terms of their size. So this is one of the special anatomical specializations of the nervous system to do this task, right? This is an example of that specialization. And I want to show you a physiological specialization as well. And to do that, I'm going to show you a comparison of responses from one of these bushy cells, right? With the responses of the auditory nerve that is the input to those bushy cells. Right? So we're going to compare the timing between the auditory nerve and the bushy cell. I've already shown you you've heard examples of the auditory nerve timing. I'm going to skip this. This just shows you, the penetrations on which these recordings depend. So here are the results. So here you can imagine that we deliver a tone, repeat it 200 times, synchronize the spikes on the onset of the tone, and we're recording from the auditory nerve. We get these dot rasters that look like this. The fact that they are in vertical rows is a reflection of that phase locking, the preference to respond at a particular phase of the response. And if you do exactly the same experiment on essentially the same frequency cell, you can see that the timing in the bushy cell is much more precise, right? The fact that these columns line up much more precisely than they do in the auditory nerve. And you can quantify that by doing period histograms. But I think it's obvious just by looking at the histogram. And this is kind of a example of, whoops. Yeah, so that gives you a sense of that, how those were obtained. And here you can see a quantification. So these are the bushy cells, and this is the range of they're measured by this factor we call synchronization coefficient. So auditory nerve fibers are in this range, whereas you can see bushy cells have sync coefficients that are much higher. So this is another sort of counterintuitive result, right? That the second order cell actually has better timing than that of the first order afferent that comes into it, right? So another, an example of how the nervous system has specialized, in this case, physiologically to handle this. Okay, so this is a physiological specialization. Now, so if now we record from those cells in the MSO that are getting inputs from the two sides, this shows you an example of one of these recordings. So these cells are binaural. They respond to stimulation of one ear or the other or both, right? So this shows you responses on the right of stimulation of just the contralateral ear, of just the ipsilateral ear, and these are now plotted as period histograms that tell you the timing of that. And you can see that the timing information is slightly different for the contralateral and the ipsilateral ear. If you look at it, you can see the peak, the arrow is set at the peak of the response. You can see the contralateral response is delayed slightly from the ipsilateral one, and you can quantify this by this phase metric. So the difference in that timing is approximately 150 microseconds, right? So now, so if this cell is acting as a coincidence detector, then it should respond maximally when we introduce an ITD into the stimulus that just compensates for this 150 microsecond sort of natural difference, right? And so the plot here shows you responses when stimulating both ears and when we're changing the interval time difference, right? So that's the variable that's being varied here. And you can see that the cell doesn't respond maximally when the stimuli are coincident, but rather when the contralateral stimulus leads the ipsilateral stimulus by about 100 microseconds. Right? So that's the prediction given from the monaural responses would be 150 microseconds, but we didn't actually do it and find enough steps. Right? So this is kind of the evidence that these cells are responding like coincidence detectors, and this is kind of gives you an intuitive feel for that. If you imagine this spike train coming into one ear, this one coming into the other ear, and we're varying the ITD, then when these two spike trains line up exactly, then you'd expect to get the maximal response, right? And then if you shift them out of phase, then the response will go to zero or near zero. And this also illustrates why you get the cyclic response, because you shifted one full cycle, then again, they'll line up, right? Okay, so this is a way of, another way of saying the same thing. This is, so what I've talked about so far are responses to tones, right? Pure tones. But of course, we hardly ever hear pure tones. So this is using sort of random noise. And what it shows you is that these cells are really computing a cross-correlation of the signals to the two ears. And this is interesting because, well, another aspect of this is that, the Army and Navy, of course, are very interested in being able to detect airplanes and submarines in open ocean, right? So engineers have developed methods to do that. And the way the Navy, the instruments that the Navy uses to track submarines in open ocean is to put two detectors wide apart, miles apart, and take those signals and do a cross-correlation of those signals. This mathematical computation. And this is kind of illustrated by this cell in the MSO. And what's shown here is that we're changing the degree of correlation of the noises to the two ears, right? So if we use the identical noises to the two ears, and they're perfectly correlated, right? But we can introduce other noise in varying amounts, to gradually decrease the correlation of the two ears until we have two completely independent noises to the two ears. Then the cross-correlation of those signals would be 0. And what you can see here is when the noises are highly correlated, 1.0, you get the maximal response. Now as you start to decorrelate the noise to 0.7, to 0.3, to 0, then the cell stops responding, right? Stops showing the peak. And in fact, you can negatively correlate by delivering noise A to one ear and then deliver minus A to the other ear. And when you do that, you can see you get an inversion of the response. That's the one shown in red here. Right? So this is just evidence that these cells are in our brains are computing what's essentially a cross-correlation of the signals, just like the engineers designed to detect, submarines in open ocean. Okay, I just wanted to show you briefly so we also do behavioral experiments, and our experimental animal is cats. Cats were picked because several reasons. One, feral cats in the wild are nocturnal. They come out at night because the prey they're after, rodents, are active at night, right? So they are really good. Obviously at night, the visual input is diminished, so they have to rely on their auditory system. So they're really good at sound localization. And their auditory system is very similar to that of humans, right? So what we learn from cats is pretty much, I think, applicable to humans. So this is an experiment just to show you that we can teach these cats to localize sounds. So what the cat has to do is to stand in a particular spot. His head has to be in an imaginary box, and you'll see that it's hard for him to know exactly where that is. So when things aren't going, when something doesn't start when it's supposed to, he starts moving his head back and forth because he's looking for that place to put his head. And then in this particular case, a light will come on. He'll have to look at the light, and then the light will go off, and at the same time a sound will come on, and he has to walk to the locus of that sound and press a lever to get a little food reward. So the light comes on here, and you'll hear the solenoid when he gets a reward. So he's licking the tube, and it's completely self-paced, right? So you'll see he then has to come back to the start point. But first, a little house cleaning. So, in my career, I also had a lot of pushback from animal rights activists. So I was a poster child for PETA. I don't know, some of you may remember the buses. Those are my cats on those buses. But, PETA claimed our cats were mistreated and abused. And, I think this illustrates their pretty good shape. This cat was actually in the lab for 13 years. Okay, anyway, so let's move on to the sort of demos that I asked you to look at. And this involves another aspect of binaural hearing, which I think was unexpected. So if you think about, so these are so-called HRTFs, which is just simply recordings made near the eardrum of the left and right ears for a transient sound, a click stimulus at, in this case, 5 different positions. So the left ear signal is shown in blue, the right ear signal in red. And these are the recordings made from the microphone. Here we've done a Fourier transform of those signals to do what the cochlea, mimic what the cochlea does. And here you can kind of see that when the sound is opposite the left ear, then the left ear signal is louder, right? The amplitude is higher than the right ear signal, and it occurs earlier, right? That's the ITD, right? And in the cat, the maximum ITD is around 300 or 400 microseconds. When the sound is opposite the right ear, you have exactly the opposite effect, right? When the sound is straight ahead, then the signals to 2 ears are approximately the same, so the ITD goes away, and the sounds have essentially the same amplitude. Right? So this shows you these cues, ITDs and ILDs, right? And you can see the ILD cue is actually a very complicated function of frequency. But we know from personal experience that we can also tell not only whether sounds are left or right, but also whether they come from above or below, right? And if you think about sounds that are right on the mid-sagittal plane, then these interaural cues, ITDs and ILDs, cannot be playing the major role because the input to the 2 ears are going to be the same, right? And that is illustrated here for sounds right on the mid-sagittal plane. The left and right ear signals are essentially identical, right? So what is the cue that is used? Well, it turns out if you look at the spectra of the plots, you can see this prominent notch that as you move down in position, that the placement of that notch drops in frequency systematically. Right? So it's thought that it's the spectral content of the signal that gives us a cue whether the sound's above or below. Right? And at this point in class, I usually do a demo. I don't have time to do it now, but I invite you to do it at home. So the demo is really easy to do. Right? Is you distort these. So these spectral cues are a product of the filtering properties of the external ear, the pinna, and the head and shoulders, but primarily the pinna. Right? So the demonstration is to show that we mislocalize sounds in this dimension if you distort the pinna cues. And the easy way to distort pinna cues is to fold your pinnas forward like this, right? If you do that, you can still hear well. But if you're trying to localize sounds, you'll still be able to tell left and right in general, but you'll fail on the up and down dimension, right? And this is very counterintuitive, right? This is telling you that the sound itself is telling you something about where it comes from. You know, that logically that doesn't make sense. So the usual explanation is that natural sounds rarely have this very narrow notch, right? That's a very unnatural sound. So we seem to store a library of those notches, and it tells us whether sounds are coming from above or below. Now, another aspect of these HRTFs is, so if you put on headphones and think about where does that sound come from when I'm listening to on headphones? Well, if you think about it, it actually seems to be coming from kind of the middle of your head, right? Which is, of course, a very unnatural spot for sounds to originate. Right? So it turns out that if you incorporate these filters, these HRTF filters, into the sounds, you can experience an externalization of the sound, right? And so I don't know how many of you downloaded the files that I suggested. Some of you did, but so at this point you can. So this is kind of virtual reality, right? You could at this point listen to this virtual barbershop, and if you didn't bring a listening device or headphones, you can do this at home. Just Google virtual barbershop, or if you access this link which Tom put on the email, you can also get it from the UW box. And so basically, what that demo does is it simulates a barbershop experience, because in a barbershop they can clip all around your head, right? So you hear the scissors go around your head. But pay special attention to whether the sound appears to be externalized, right? And I think that's a very unusual experience. So I actually had, when my grandson was 2 or so, he was, on the computer, so I thought, oh, well, let's have him listen to the virtual barbershop. So I should have videotaped it, because, when I started it, he immediately turned and looked because he thought somebody opened the door and came in, because that's how it starts out, right? It sounds like somebody's out there. And then they go through a scenario where at some point the guy says, okay, I'm going to put this over your head, you know, because we're going to start the cutting your hair. And my grandson immediately said, no, 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 and, you know, tore out the headphones, and then refused to get a haircut for about a year or so. His parents weren't too happy about that. So this is another sort of interesting aspect of, which was, I think, unexpected when people started studying these HRTFs and incorporating them into signals. They found, you know, you could actually produce this externalization of sound. And of course, everybody's pinna filters are different because our ears are different. Sizes are different, shapes are different, right? So you're, in these examples, you're essentially hearing through somebody else's ears, right? And so for some people, it's a very good, sounds very, externalizes very well. And other people probably, maybe not so much, probably because we're not using your own HRTFs, and we know that you get used to your own HRTFs, right? And so this is I think probably the most fun example. There are another two examples. And by the way, these studies of the effects of HRTFs were actually largely done here at UW in the psychology department by Fred Whiteman and Doris Kistler, who have now moved to Tennessee. But a lot of the HRTFs that are online are using recordings they made in probably UW undergraduates. And then let me just close by a very short explanation of something which is known as a binaural masking level difference, which is another aspect of this. This is a phenomenon, and the fourth of the examples is an illustration of BMLDs. And the demonstration runs something like this. So imagine you put on headphones and deliver a signal, which is the tone, and some background noise to the same ear, right? And then you adjust the level of the tone until you just can't hear it. That's your threshold for detecting the signal, right? And in the demonstration, what they do is they gradually decrease the tone in steps. And if you count the number of steps you can hear, then that gives you a sense of the threshold, right? So if you do that monoraily, with both sounds to one ear, the guy is frowning because he doesn't hear it very well. It's hard to hear the signal, right? Now, add noise, right? We're trying to hear the signal, but now add noise, but add it to the other ear. So we're adding the same noise to the other ear. And that immediately makes him smile because he can now hear it better, right? So then we're trying to detect the signal, remember? So now let's add more signal, but add it to the other ear, right? And when you do that, the sound becomes less detectable. Very counterintuitive, right? You can't detect it as well. The guy's frowning, right? And now, take the signal and invert it. Just multiply it by -1, right? Or you could, physically, you can just reverse the terminals, connections on the speaker. And when you do that, the subject is happy again, right? So this is, and you can get a almost 15 dB improvement in hearing the signal to noise by doing this simple procedure. And this actually has an interesting history, right? The discovery of BMLDs was made pretty much simultaneously by two investigators. One of them was working for the Air Force during World War II, right? So during World War II, he was a Harvard psychologist, but during the war, everybody's working on a war effort, so he went to work for the Air Force. The Air Force put him on the project of making sounds in the cockpit of fighter planes more detectable, right? You can imagine hearing in a loud cockpit of a fighter jet, a lot of noise. You're trying to listen to the conversation over headphones. You know, pilots are having a lot of trouble, so they put him on this task. So he discovered that if he simply took the headphones and inverted, you know, cut them and soldered them back in reverse order, he got a 15 dB improvement in the signal noise ratio, right? Simple procedure. Didn't understand why, right? But this spurred there are probably 200 to 300 papers on BMLDs because it's such a non-intuitive result, right? You add signal, the noise it becomes less detectable. You add noise, it becomes more detectable. You know, it goes against all your sort of intuitive notions. So if you you know, you can listen to this series goes through those 4 steps in the order that I showed them. Okay, it's 8 o'clock. I've probably gone over my time, so thank you for your attention. And we're happy to answer any questions. Yeah.
Can this process be used in hearing aids?
Yeah, I don't know of any hearing aids that actually incorporate it. You know, often hearing aids are independent. You know, they're not coordinated. That's actually one of the problems with cochlear implants, right? So if you know the next step beyond hearing aids, if you go deaf, you know, one of the cures is now cochlear implants. But binaural cochlear implants, you don't gain this advantage because the two implants are independent. So in order to get that sensitivity on the microsecond level, right, you need coordination between the two devices.
Why don't they have it?
Well, it makes the cochlear implant much more cumbersome. So it's a hot area of research. So actually, there's a lab on campus that's their primary field of study. And at the moment, cochlear implants are kind of either inside your head or on a little device that hangs off your ear. But in order to coordinate them, you have to carry this little box, right? So it's very cumbersome at the moment. So a lot of people are working on ways to improve that. Yeah. Probably it will come eventually. Yeah.
You mentioned that the hair sizes depend upon the frequencies you want to detect. Does that mean that bats and dolphins have very small hairs, and elephants may have very long hairs?
Sorry, so you think bats?
Bats and dolphins may have very smaller hairs than we have for very high frequencies.
Right.
And elephants may have very long hairs for very low frequencies, which they...
Yeah, it's not the length of the hair that's important, right? So it actually turns out it's the stiffness of the basal membrane that sets the frequency tuning from low to high. And those animals, right, have cochleas that are highly specialized, right? Especially bats that hear up to 200 kilohertz or so. Yeah. Yeah.
When you talk about adding noise and analog to digital converter, they call that dithering, but you have to sacrifice dynamic range. You sacrifice any dynamic range when you add noise to
well, in this experiment, we're adding noise to make it less, to test how detectable the signal is, right? We're not adding noise for, you know, it's just an experimental procedure. I don't know if I answered your question. Yeah, we're not really dithering this signal. I mean, we're not adding noise to intentionally dither the signal. Yeah, Tom.
Kind of an old stereo question, but my understanding is when Bose came out with one of their speaker designs for that little thing, one way that they made it sound better was to have the sound travel through a pipe that's like 3 or 4 feet longer. is that kind of offset the kind of thing that your 2 ears are hearing to get a better,
Yeah,
richer
it's
sound?
sort of related, but not, so they're trying to recreate, right, in the sort of concert hall experience by having, you know, trumpets on one side and the strings on the other or something like that, right? So to do that, one of the tricks in the trade is to delay inputs from on one side or the other by a small amount, and that's kind of one of the techniques. Not quite the same as what our heads are doing in computing that delay.
Then, I go to EAA every year and they have some headphones like I think you're talking about starting in World War II. Can you tell us more about the sound cancellation or noise cancellation technology? How does that work? On the
Yes, well, so this BMLD effect, right? I should have mentioned it. So, you know, we normally study sound localization. That is, how do we tell where the sound comes from? But that ability is exactly the same as what's often called the cocktail party effect, right? So imagine yourself in a crowded room, cocktail party, right? And you're trying to carry on a conversation with somebody. Your ability to do that in the face of a lot of noise coming from all the other conversations in the room relies on the same mechanism. That is, it relies on the fact that the speaker you're trying to listen to comes from a particular point in space. The noise is coming from elsewhere, just like in the cockpit of the pilot, right? The noise is coming from all over. He's trying to hear through the headphones now. So the psychophysical experiment is if you took a dummy head, you know, and put it in the middle of a cocktail party and recorded from the left and right ears of this dummy head the cocktail party, right? Then if you played back what you recorded through one, you know, just through one microphone, just through one ear, then it's very difficult to follow any conversation that you can hear because it's all jumbled, you know, you've got all this noise. But if you play it back listening to both ears, right, then you can do what we do in a cocktail party, which is to listen to this conversation or switch our attention if we hear our name over here, right? We can adjust our attention, so to speak, and that ability also is essentially the same thing that what that fox was using, you know, to localize that bull under the snow. So, you know, although we don't, I would say, don't use sound localization, certainly not to survive in modern our lives, but we do depend upon it to do this signal and noise problem. You know, we're in a crowded room, and you probably, you know, my hearing, there's a natural loss of hearing with age, so I've actually just went to my ENT, and he says, well, it's ready for time for hearing aids. But you know, people who have loss of hearing, usually the major problem is in a noisy environment, right? They can still carry on conversations on the phone in their bedroom, but put them in a noisy cocktail party, then things get really difficult, right? Yeah.
I have one comment and then a question. And so my wife said that the reason we have two ears is because it goes in one and out the other.
I've discovered that in class as well.
The question I had is there like the sensitivity is the basilar membrane, the length of that?
The width, actually.
The width.
So in the movie, you might have noticed that it's wide on one end and narrow on the other, right?
So is there evolutionary record that shows that both prey and predators are having increased width over time and as a survival mechanism? something like that?
Well, people are using, you know, the fossil records of skulls to make deductions like that. I don't know. I don't think, you know, the first of all, of course, all animals have a different range of hearing, right? Which has probably evolved for a number of different reasons. I mean, we have relatively low frequency hearing. That is, you know, at birth we can hear up to 20 kilohertz, but you know, I probably don't hear over 8 kilohertz or so. But of course, the main thing we use hearing for is communication, right? So speech sounds are primarily a low frequency. They have primarily low frequencies. So the high frequencies are not critical for speech. Whereas, you know, bats that are using echolocation, they have to be able to hear those high frequencies, right? Because they use very high frequencies because the things they want to echo off of, moths and insects, are small, right? So the wavelengths have to be small in order to bounce that echo back. So I would say, you know, there's a large variety in the you know, the basilar membranes of different animals, but they're probably largely due to their particular behavioral niche, right? And what they use.
Be like there'd be a survival benefit being able, if you're a predator, that the better you can localize your prey, the more likely you're going to survive. And as well for the prey, the more likely you can attack your predator.
Right.
That you're going to, and so that there'd be that survival.
Yeah. Well, there is, as I argue, and it's more of a problem for the predators than for the prey, because so the model animals that have been used to study sound localization are all predators. So barn owls catch rodents in the dark, humans, cats, ferrets. These are all predator animals. They're really their acuity, sound localization acuity is very high. You know, mice, rats, the prey they're hunting, they're relatively poor because they didn't have, you know, behaviorally, they don't have to tell whether that predator is at 10 degrees or 20 degrees, right? They just have to know it's over there somewhere. Whereas the predator has to be very precise. Yeah. Yeah.
Two thoughts. First, you complicated my thinking about what it's like to be at a symphony concert, and it's complicated my thought about how the instrumentalists hear each other. And I'm now more amazed than ever that they ever play together. Cats have ears that are here. We have ears that are here, so do owls. But cats have very flexible, thin pinnae. Is that what you
Pinnae, yeah, pinna.
Pinnae.
And they can move them,
And they move.
which we,
Does that make up for some of the fact that they're up on top of the head and give some ability then to?
well, you know, I don't know if the top of the head and the side of the head makes much difference, but certainly being able to move them, I think, is a feature which.
Straightforward. They don't seem they have much relative to the head. It seems like they're not as different as ours are. There's not as much distance of head between the ears.
Well, of course, their heads are smaller.
Yeah.
But we've actually also studied pinna movements in the cat, right? By behaviorally, we can measure their pinna movement. So it turns out when they're put in a task where they're asked to tell us where a sound comes from, they will rotate their pinnas actually before they move their heads, probably because the moment of inertia, it's easier to get the pinna moving than the big head. And then, interestingly, so if a sound is turned on to the left pinna will orient to it, then the head will start to move, and the left pinna in space will then counter-rotate to compensate for the head movement. So in space, the left pinna will stay in the same spot, even though the head is making a big turn. So, you know, this is obviously something that's been designed into the system, and it's very much like, actually, we have a reflex like that in the visual system. So we have a so-called vestibulo-ocular reflex. So when I turn my head to the left, both eyes reflexly move to the right. And that's to try to keep the retina pointed at the same point in space. Right? So you can demonstrate that by shaking your head violently like this, and things can stay relatively stable. Or another way to think about it is, think about if you're out jogging or riding a bike or riding a horse, right? Your head is doing all this, bouncing around. On the retina, you would think that the visual world would be doing that too. But no, the visual world is pretty stable because the VOR compensates for all of your head movements. We move our head to the left, VOR points our eyes to the right to compensate for that leftward head movement. So the pinna does the same thing for the acoustic world. Yeah.
55 years ago, I had a baby in June, and in August I had a stroke in my left ear. I thought it was a baby falling out of bed, but nobody cried. And I lost the sounds above the sound of speech. I'm eligible for hearing aids now, but I don't always wear one. I only have one. I turn my head for localization.
Right, because you have a good ear and a bad ear, right? So naturally, you've learned to point your good ear at the sound, right? Now, you know, when you do this in animals, right, there are some compensations that the nervous system will do, as you might expect, to compensate for that, right? But it can't compensate completely. And so particularly low-level sounds, faint sounds, you need to point a good ear towards it to hear that.
I never thought about it until I heard you say this presentation.
Yeah. Yeah.
I noticed in that video of the fox that it seemed to be utilizing multiple inputs by moving its head around. So he's getting a lot of information on localization.
Right, it's doing this. Right?
And not moving the ears, but moving the head.
Yeah.
Like this,
right.
like this, a little bit.
Yeah, so, you know, of course, one of the important parameters for the fox is how far away is it? You know, this distance, right?
Yeah.
What I talked about so far, we haven't considered distance as a variable, right? So how do we judge how far away a sound is? We actually don't know much about that.
Well, we take multiple points of
right. Well, if you look at it from different ways, you can triangulate.
Yes,
that helps, right? But actually, it looks like one of the biggest cues is the intensity of the sound. We have some notion about how loud these sounds should be, and if it's very faint, then we think it's far away. If it's if it's loud, we think it's close. But of course, you can fool, and in psychophysical experiments, you can fool a subject easily by playing around with that.
That fox was not fooled.
No, right. Yeah, so in a way, he's doing this and that to sort of triangulate the distance too. Yeah. Yeah, it's really remarkable that they can do that, right? Yeah.
Going back to the topic of cochlear implants, I've heard that the senses that the implant gives to the brain is quite different than our natural ears. And could you talk a little bit about what that is like to experience that before the patient learns how to interpret those new senses?
Well, I have a demo. Do you want to hear it?
Yeah.
So I think to answer your question, I think it varies a lot patient to patient, right? So a lot of deaf patients are born deaf, or they've never had hearing experience. For them, hearing, trying to interpret speech through a cochlear implant is usually difficult, right? But then there are patients who have, suffered deafness after they've heard, after they've lived a life with hearing. In fact, one of the primary causes of hearing loss is antibiotics. So if you're taking antibiotics, especially high levels, high concentrations, it can destroy those hair cells. So I'm trying to look for this file and talk at the same time. There's a professor at Purdue, physics professor, who became deaf late in life. He was, oops. I'll just say I can't do two things at once. Anyway, so he studies high energy physics, right? He's a professor at Purdue. So actually, he got cancer, the way he tells the story. He got cancer when he was about 50 and had to take high dosages of antibiotics for the cancer. And that caused him to lose his hearing, right? And after that happened, he learned about cochlear implants and decided he wanted to get one. So he had cochlear implant surgery and was so enthralled by how well it cured his hearing that he now goes around the country giving 2 lectures, one in physics departments about high energy physics and another on cochlear implants. And the relevance to what you're asking is that he said that the moment they turned on the implant, he could understand what the nurses and doctors were saying. So he immediately, there was no transition. But that's not always the case, particularly for people who don't have hearing experience.
Is there such a thing as a lazy ear, like there's a lazy eye, and one ear is occluded for sound during the early development that the brain doesn't develop for the hearing?
Lazy ear. I've never heard of that.
Lazy eye, if you occlude.
Yeah, right. I know the lazy eye problem. As far as I know, I've never heard of that. So it's kind of the loss of hearing in one side, I guess. Right? Okay, so these are simulations. These are attempts to simulate what a cochlear implant patient hears through the cochlear implant, right? And modern cochlear implants have 22 channels. That's the standard cochlear implant now. But in the early days, they put in, when they were developing them, they first started with a couple channels. So what the example here is examples of 1, 2, 8, 4, 8, 16 channels of a speech sound, right? So here's a 1-channel example. So your job is to raise your hand when you can understand what's being said. Nobody? 2-channel? Four-channel? 8?
I like to play tennis.
16
I like
channel?
to play tennis.
And here's the original.
I like to play tennis.
So many of us heard I'd like to play catch, though, at 8.
Right, so you didn't quite get it all, right? So he said that he can't tell the difference between the original sound and the 16-channel sound. It sounded exactly the same to him, which suggests, I always wondered exactly what you asked. What does it sound like? And so this suggests that at least the simulation may not be perfectly correct, but it's probably close. And here's what music sounds like. Anybody recognize that? What?
It's the sugar plum fairy.
Yeah. Good. Well, you got the rhythm, probably. But, you know, as you can appreciate from that, one of the things cochlear implant patients complain about is music doesn't sound good anymore. So the ability to reproduce the pitch of sounds is not done very well. Yeah.
Do noise canceling headphones incorporate some of the principles you talked about?
Not really. So what the noise canceling headphones do is they have a microphone that picks up the external noise, right? And then quickly computes, essentially reverses that noise and plays it back in the ear to cancel the external noise.
So for purposes of blasting your hair cells, if you're hearing blissful silence in a noisy environment, you're just being fooled into thinking that's blissful silence, and you are broken your hair cells.
No. You are the cancellation occurs out in the pressure waves so they would be good, yeah. So you know, if you're particularly on airplanes where you're sitting there in that noisy environment for long flights, that's a good thing to do. Yeah. Yeah.
What was the animal on the right in the original?
I was afraid somebody would ask that. I'm not sure. It might be a, what is it, panel fox? What is it? Phoenix. I think, but I'm not positive. I think I just Googled pinna. Came up with that.
Any other questions? If not, thank you very much.